Hello everyone, my name is Jordan Metcalf, and this is my 3D model of a eukaryotic animal cell. I chose to make this out of Lego because I thought it would be an interesting take on it, and it was really cheap. So over here, I have a little kind of reference kind of table to kind of show what colors I chose to associate with which particular part of the cell. So red, this bright red, represents the plasma membrane. Purple represents the nucleus. The lime green sphere or half sphere is the nucleolus, and I chose dark gray to represent the rough and smooth endoplasmic reticulum. There's a soft panel on this one and no soft panel on this one to differentiate between rough and smooth. And I chose just a lighter gray to represent the Golgi apparatus, as you can see right here. And I chose both yellow and brown to represent mitochondria. That's actually roughly what it's roughly the colors that it's depicted as in the book. And for the ribosomes, focus. It's not focusing. Well, anyway, we'll just have to do with it. There it goes. For the ribosomes, I chose to go with the golden one studs, I guess. You can see right there. And for the lysosomes, I chose the two colors of a very light, almost aquamarine blue, and just a regular tan. So right there. And for the flagellum, focus. For the flagellum, I chose the tubes to be represented by the white, and the outer layer to be represented by the maroon red. And for the extra credit, I chose the kind of neon red to represent the chromatin of the inside of the nucleolus. And if that doesn't work, then I chose the lime green to represent the centriolus that I added in. All right, and so here it is. Let's start. So there's the red plasma membrane all the way around right there, and the plasma membrane acts as a boundary between the inside and the outside of the cell, trying to make it as circular as possible. And then there's the purple nucleus right in there, and the nucleus is a compartment that holds genetic material within the eukaryotic cell that or rather the chromosome of the cell, which contain hereditary information. Moving on to the nucleolus, which is the green sphere. The nucleolus is a type of RNA, and it's called lowercase rRNA or ribosomal RNA, and that's what it produces. And so then we move on down to here to the rough and smooth endoplasmic reticulum. Right here, as you can see, the smooth endoplasmic reticulum has the smooth covering, and the rough does not. And the rough endoplasmic reticulum is studded with ribosomes, right here, as you can see, the gold studs, on the sides of the membrane. And while we're here, the ribosomes, they are composed of two subunits, one that is larger and one that is smaller. Let me stay on that. And the ribosomes act almost as a workbench, and there the information contained in the RNA from the nucleus actually is contained from the nucleus is used to synthesize a polypeptide chain. So there's the ribosomes right there while we're at it. And so, yeah. And then I'm afraid I didn't talk about the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, right along here, is continuous with the rough RNA, so as you can see, they're attached. And but the only difference really being is that it does not have the ribosomes on the end. And so moving on to the Golgi apparatus right in here, just the lighter gray. The Golgi apparatus, it consists of a stack of slightly curved yet flattened saccules, and it receives transport vesicles sent to it by both the rough and smooth endoplasmic reticulum. And there it is right there. And moving on, we did the mitochondria, which is right here, is represented by the yellow and brown, as I said earlier. And the mitochondria is really just kind of the powerhouse of the cell because of what it produces. It produces the ATP that cells typically utilize. And there it is right there. So yeah, just the yellow outer layer and the brown inner workings. And so then moving on from the mitochondria, we already talked about the ribosomes. They're right there. So let's move on to the lysosomes. Lysosome is right here. Lysosome is basically a vesicle produced by the Golgi apparatus, where the lysosomal enzymes digest the contents of the vesicles sent from the Golgi apparatus. So there it is right there, with the tan inner workings right there and the blue disc. So yeah. And then one of the last but not least is the flagellum right here, represented by the maroon red and the white tubing. The flagellum, it moves in like a snake-like fashion, because in some single-celled protists actually utilize the flagella to move around. So it's kind of a transport, almost vesicle, but it's just it's a way for single cells to be transported. And so for extra credit then, I chose to go with the chromatin on the inside of the nucleolus, which is these red dots right here, represented right there, chromatin. And the chromatin is a network of DNA, protein, and a small amount of RNA. Chromatin condenses and coils into a rod-like structure, which is called chromosome. So before a chromosome is a chromosome, it is at first it is a chromatin. And if that doesn't work, then I decided to add the centriolus right here, represented by the neon yellow. And the centriolus is located in the centrosome, and it is a short barrel shape. So I try to get the barrel shape right here. Organelles, they're organelles really, and they're composed of microtubules. I don't know how to say that. And so yeah, there it is. And that's my whole 3D model of the eukaryotic animal cell.